we should go ahead as scheduled and actually present our new, our two speakers now. Dr. Wen is an assistant professor at the Department of Psychiatric and Behavioral Science at Emory University. He will be talking to us about the brain organoids and how they could help understand and treat GRIN kids. And then we'll follow with Dr. Amy Ramsey. I think everybody knows her by now, but she's from the Department of Pharmacology and Toxicology at the University of Toronto. And she will be talking about the mouse model and the use and its use for studying GRIN disorders. So I'll leave it to you guys. Thank you very much and enjoy the talks.
Hello, everyone. Thanks for organizing this meeting and inviting me to give a presentation here today. Let me share my screen. Yeah. So the research of my laboratory mainly focus on understanding the disease the mechanism underlying the neurodevelopmental disorders. In particular, the intellectual and developmental disability. And we know that we call the IDD, such as autism, ADHD, and intellectual disability, affect millions of kids worldwide. And it affects about 10% of children born in United States each year. And we can see that the number keep increasing in recent years. For example, for the autism, 1 in 68 kids has been diagnosed as autism, and there's 30% increase from 2008 to 2010. And the challenging is that effort to develop therapeutics has been challenging, and very few novel treatments has been developed in the past decades. And to develop new treatments, we need to have a better understanding of the disease mechanisms. But how we can do that? So these are 3 major preclinical models that are widely used in the laboratories, including the postpartum brain, the brain imaging, and animal models. All of them has already provided very useful information for our understanding of the disease, but at the same time, they also have some limitations. For example, the postmortem brain, they present the endpoint of the disease and cannot tell us how the disease initiate and how the disease progress. And the brain imaging also provide very useful information at the system level, but they provide limited information at the cellular and the molecular levels. And animal models are widely used in the laboratory, and they also have already provided us many insights into the disease biology. But we realized that there are fundamental developmental, biochemical, and physiological difference between animals and humans. And so far, for many cases, or maybe for most cases, it has been failed to translate the basic finding into the human therapeutics. So we are wondering whether we could have a human-specific model that we can use the model to monitor the disease initiation and the disease progression for the neurodevelopmental disorders. So one approach is the human-induced pluripotent stem cell, or called the iPS cells. This technology was first developed by the Nobel laureate Dr. Yamanaka in 2006 in mouse and 2007 in human. Basically, by introducing 4 transcription factors, now we call them as the Yamanaka factors, into the human somatic cells, like the skin fibroblast, and this will reprogram this fibroblast into the human embryonic stem cell-like cells, and we call these cells the induced pluripotent stem cell, or iPS cells. And basically, right now, we could collect the samples from the patients. For example, the skin fibroblasts, the blood samples, or the urine samples. And then by introducing the Yamanaka factors, we could reprogram these somatic cells into the iPS cells. At the same time, if we know the genetic mutation in these cells, we could do the gene editing by the CRISPR-Cas9 to correct the mutation. Or at the same time, we can also introduce the same mutation to a healthy control background. And this iPS cells called the isogenic lines. And then you could differentiate these iPS cells into the potentially any cell type we want, for example, neurons. And from this neurons, this patient neuron, we can see what's going on in these neurons and what's the phenotype. And by identifying this phenotype, we can also use this phenotype as the readout to set up a high throughput screening assay to screen novel compound that maybe can use back to the patient for the treatments. So compared to the other model system, the human iPS cells have several advantages. So first, these patient-specific iPS cells carry the exactly same genetic information from the patients. So this is extremely useful to study the complex human disease, like the most of the neuropsychiatric disorders that are we believe that they are the polygenic disease. And also, these iPS cells are multipotent, means that potentially they can give they could be differentiated to any cell type we want, especially those cell types has the disease relevance. For example, neurons or glial cells that we believe that have the relevance to the neurodevelopmental disorders. And these iPS cells also are renewable, so means that we potentially have unlimited source for the disease study or for the drug screening, or in the future, maybe for the cell replacement therapy. And they can also easily be accessible. We can generate the iPS cells from small pieces of the skin biopsy and also a small amount of whole blood samples or the urine samples. And there's no age limitation. We can generate the iPS cells from subjects from patients that at any ages. And importantly, as I mentioned, that they are the patient-specific, then they could be very useful for the future personalized medicine, for example, for the personalized drug screening. And also, there's minimal ethical issue, and there's no immunogenic issue that so that the cells differentiate from the iPS cells could be used for the future cell replacement therapy. But compared to other model systems, the iPS cells also has some limitations. Definitely, we can see that this is the in vitro model, that we cannot do behavior tests on this model. And also, the iPS cells, the cells differentiate from the iPS cells somehow still less mature than the cells in vivo. So, and also they are the neurons, for example, they also lack of myelination. So this also one of the limitation currently. So in the recent years, many differentiation protocol has been established to differentiate these iPS cells into the many cell types in the human brain. For example, from the iPS cells, we can differentiate to the neuroprogenitor cells, brain region-specific neuroprogenitor cells, and then we can differentiate them to different type of neurons in the brain. For example, the cortical glutamatergic neurons, hippocampal neurons, and the GABAergic interneurons, as well as other type of neurons, for example, the dopaminergic neurons and the motor neurons. At the same time, we can also differentiate these iPS cells into the glial cells, including the astrocyte and the microglia. For example, this one example of the cortical neurons differentiate from the iPS cells. We can see that they can form the synapse in the in vitro culture, And they can also have the spike structures that similar to the in vivo case. And importantly, they also could respond to the stimulation, like the potassium chloride stimulation that indicated by the calcium imaging. And when we do the patch clamp recording, we can see that they can fire action potential and have the spontaneous synaptic event, synaptic activity. However, this is the 2D differentiation, and this 2D culture state lack a brain cytoarchitecture. They also lack 3D microenvironments, and this 2D cultures, although sometimes we can do the co-culture with different cell type, but they are missing many cell types when compared to the human brain. And we know that the human brain is a 3D structure. So we are wondering whether we can have a model that can recapitulate the human brain structure. And in 2013, Lancaster first developed the 3D brain organoid from the human iPS cells using the bioreactor, spinning bioreactor. And we can see that they have very similar structure that compare to the human cortex. But at that time, this is the first generation of human brain organoid, and we use the traditional bioreactor. So the limitation is that this throughput is low, and the cost is quite very expensive. And also, this is a generic brain organoid that have different brain regions here. So when we use this brain organoid for the experiments, there are lots of the variability between organoid to organoid. So in 2016, when I was a postdoc at Johns Hopkins, working with Dr. Hong Jin Song and Guo Ming, the student in our lab, Xu Yu, he developed miniature bioreactor by the 3D printing. And here is what the brain organ looks like in our incubator, that we can see that with this miniature bioreactor, we can use the traditional 12-well plate, and then we can grow the organoid with many conditions. Then we can test, we can compare different treatment conditions at the same time. And we also modify the protocol to generate the brain region-specific organoids, for example, cortical organoids or the midbrain organoids. And here are the cortical organoids look like in the culture plate. And when we zoom in, we can see that this organoid have different layer structure that's similar to the human cortex, including the ventricular zone, subventricular zone, and the cortical plane. And when we look at the development of this human brain organoids, we can see that they also resemble the human brain development. For example, at the early stage, that the majority of the organoid are the SOX2 positive ventricular zone-like layer. And later on, for example, at the day 56, we can see that the neuroprogenitors from the ventricular zone start to migrate out to form the cortical plate and differentiate into the cortical neurons that are CTIP2 positive. And even at the later stage, like at the day 84, we can see that in the cortical plate, they can also form different layers as the human cortex. For example, the CTIP2 and the TBR1 positive dip layer, as well as the SEPI2 and the CUS1 positive upper layers. And importantly, in this human brain organoid, we have the primate specific layer called the outside subventricular zone. And this is not exist in the rodent models. And this layer also very important for the neurogenesis in the primate, for example, the human brain. And we are wondering whether this human brain organoids also recapitulate the human brain molecular signatures. To confirm that, we did the RNA sequencing on the brain organoids and compare to the transcriptome from the human brain. And we can see that this brain organoid mainly recapitulate the molecular signatures of human brain. And when we look at the different stage of the human brain organoid, we can also see that this organoid resemble the human brain development. For example, at the early stage of organoids, like day 26, it's like a fetal brain in the first trimester, and around day 50, the organoids look like the fetal brain at the 8 week to 12 week after the pregnancy, and the day 100 is about a fetal brain in the second trimester. So with this brain organoid, we could use them for the disease modeling, and we can also use them for the drug development, and in the future, we can also use them for the personalized medicine, identify biomarkers, and also assess the environmental risk factors, and study the neuroimmune interaction, as well as to study the neuro-oncology. And here I want to give you guys an example how we use this organoid to study the disease. So I teaming up with a colleague at Emory, Dr. Tang Jin, that we generate the iPS cells from the kids with the Fragile X syndrome, and then we make the organoid from these iPS cells, and we identify that in the Fragile X organoids, they have the early differentiation phenotype that when we compare the organoid at the day 56, we can see that Fragile X organoid exhibit accelerate cortical layer formation. And we also observe that the Fragile X organoid also have increased number of the synapse when compared to the control organoid, suggests that they also have the accessory synapse formation in this Fragile X organoid. And then we also do the patch clamp recording to look at the neuronal functions in the Fragile X organoid. And we mainly focus on one type of the cortical neurons, that is the CT2 positive cortical neurons. And we can see that in the Fragile X neurons, they fire more action potential when compared to the control, suggests that they exhibit the hyper-excitability. And we also generate the iPS cells from the GRIN kits. And this is one example that the organoid differentiate from the iPS cells with the GRIN1 G620R mutation. And we also observe that the GRIN organoid neurons exhibit decreased synaptic activity when we record the spontaneous excited postsynaptic currents called the spontaneous EPSC. And we can see that both of the frequency and amplitude are decreased in the GRIN organoids. And to gain the molecular insight into the organoids, we could do the RNA sequencing and do the transcriptome analysis. Here is the data we from the Fragile X organoids, and we can see that loss of the FMRP leads to altered expression of genes that are involved in neurodevelopment, including regulation of neurodifferentiation, axon guidance, chemical synapse transmission, and social behavior, synapse organization, and many terms that involve in the neurodevelopment, and for example, the neuromigration. So we can see that from this RNA sequencing, maybe we can identify the biological pathway that underlying the pathogenesis of the disease, and those biological pathway could be the potential therapeutic targets for drug development. And to look at the transcriptome alternation at the single cell level, we also did the single cell RNA-seq on the brain organoids. And here we can see that we identified many cell types in the brain organoids. And we can also identify the gene gene expression alternation in each cell type. For example, in the neurons or the glias. And we can also compare the case and the control. We can also identify whether there's the mutation, the genetic mutation will affect the specific cell type differentiation. And for example, in this Fragile X organoids case, we can see that they have less neuroprogenitor cells, but they have more differentiated neurons, suggests that they have the early differentiation phenotype. And when we compare, when we look at some specific genes expression pattern in the Fragile X organoid, for example, the DCX, one of the immature neuron marker, that we can see that it's highly expressed in some population, but they lost in some population in other cell types in the organoids. And also, there's another marker for the neurons that called the tube 3.1, and then we can see that in the fragile X organoids that they are decreased significantly in specific cell type. And with this single cell RNA sequencing, we can also do the called the pseudotype analysis to look at their developmental trajectory from the original cells. With this pseudotype analysis, we can see that the developmental pathway for the control organoid, and this the trajectory for the fragile organoid. We can see that they have very different developmental pathway for the case when compared to the control. And we are wondering, from this data, we may be able to identify the pathway or the mechanisms underlying this alternation of the developmental trajectory at the molecular level. And the last question is that whether we could use this iPSC models or the organoid models for drug development. So the idea is that with the patient, there's two types of screening we could do with the iPSC models. First one is called the micromedicine, that we use the patient-specific iPS cells then to differentiate them to the cell type that have the disease relevant from, for example, the neurons. And then we use the patient, their own neurons for the drug screening, and this for the personalized medicine. And on the other hand, we also could use this iPS cells for the called the macro medicine, that basically we generate the iPS cells from a cohort of patients that they have some homogeneity. For example, the GRIN patients GRIN kids that they have they share some common phenotype. So the patient cohort usually used for this kind of screening need to have some homogeneity. For example, the genetic homogeneity. That one example is the GRIN, that they have the rare, highly penetrating mutations. And the other type of homogeneity is called the clinical homogeneity, that they either have the endophenotype or they have the similar pharmacological response. And with this screening for the from the cohort of iPS cells, then we may identify the new compound that can use for more patients that share some similar phenotype. And here's one example. We use the iPSC model for the drug screening. That this project is to screen compound that can prevent the Zika infection, or we know that the Zika virus causes microcephaly in the fetal brain. And at that time, we used iPS cells to identify that indeed the Zika virus mainly target the neuroprogenitors and the exercise in the fetal brain. And we also found that the Zika virus caused the cell death of the neuroprogenitors and exercise. So we use this as the readout to establish the high throughput screening assay using the cell viability as the readout, or the one of the cell death indicator called the caspase 3 or 7 activity assay. So with this assay, we have screened about 6,000 compounds, including 3 major libraries, including the raw pair library and the FDA approved drug library and the clinical candidate compound that are in the clinical trial. And from all these 6,000 compounds, we identify 35 potential hits. And eventually, we identify 3 compounds that can either inhibit the cell death or prevent the virus infection. So this is the 2D screening assay using the iPSC models. But we are wondering whether we could use the human brain organoids as the screening platform. To do that, we collaborate with Dr. Han Lu at the Georgia Tech to establish a microfluidics platform to grow the organoid and to use this platform for the screening of compounds. And this is how it looks like for the microfluidic device. And right now we are scaling up this multi-well microfluidic device to the 96 cell format or the 384 well formats for the screening. And the good thing of the microfluidic device is that they could control precisely the culture conditions. And we could also do the in situ imaging assay to monitor the organoid development and the phenotypes between the case and the control. And the device also are automatable, that in the future that we can do this screening automatically with the robot system. And they also are scalable, that as I mentioned, that right now we are scaling up it to the 96-well or 384-well prey footprint. And we can use them for the multi-compound screening. And we also could test the candidate compounds with the brain organoid. Here is one example that we test some compound for the Fragile X organoid, and we can see that we use the synaptic formation as the readout. As I mentioned that in the Fragile X organoid, they have the accessory synapse formation. And when we use 2 compounds here, one is the mGluR5 antagonist, and we don't see effects of this antagonist on the synapse formation. But we use another compound, is the PI3 kinase inhibitor. We can see that indeed it can rescue the synapse formation phenotype in the Fragile X organoids largely. So this is what I talk about today on why and how we use the brain organoid to study the neurodevelopmental disorders and to and also to screen and test potential therapeutic compounds. But at the end, I want to say that human iPSC is one of the models, but we also need to combine the data with the results from other models. From, for example, the clinical assessment, the human genetic data, the data from brain imaging, data from the postmortem brain, and the data from the animal models. And by integrating all the data from different model systems, that we maybe have a better view of the, we have a more complete view of the disease and have a better understanding of the disease biology. And here I want to thank all the people in my lab, especially Ying and Yanfei, that and Jie and Weibo involved in the Fragile X project and the GRIN project. And I also want to thank all my collaborators involved in the data I present today, especially Amy and Rob, who helped me to initiate the GRIN project. And definitely, I want to thank all the GRIN family who donate the sample to us. And I also want to thank all the funding supports. Thank you for your attention.
Thank you, Dr. Wen. If you don't mind, we just go to the next speaker, and then we do the little question and answer. So far, there's no questions. So please, if anybody has a questions, this is the moment. Thanks. Sure. Thank you. So I give Amy.
Hello, everyone. I'm just getting my, just going to get my presentation pulled up here. Okay. Oops. All righty. Can everyone see this okay? Sandra, can you see this? I hope so. Okay. Okay, great. All right. First of all, I want to say thank you so much for inviting me, and I hope that you're not getting bored of seeing me. I really appreciate that I have been able to share this with you, and I'm here to try and answer questions that you may have. And I thought that I would just talk more generally about how mice can be useful. And I think it's really nice to combine this with what we just heard from Jae-Sing Wen. And I want to say, Jae-Sing, that's beautiful work that you're doing and very exciting. So I think what I feel as a researcher is that parents and families have a lot of questions that we should be able to answer for you. And this is what we want to do in our research, and what we can try to approximate in mice. So many of you, once you get the diagnosis, you want to know, is this a gain or loss of function? Because that could one day determine what kind of medicines or therapies that that your child might get. And can we expect things to get better or worse or stay the same over time? This is a really important question for planning, and then more specific answers about treatments, like is there a certain diet or medicine that could benefit my child? This is the most important question that I think you really want to know. But there's also a lot of questions like, why is this happening? What's actually going on? And what's the root cause of these seemingly very different symptoms? And it's not satisfying just to know, oh, it's the NMDA receptor. We really want to know what's going on in the body. And I think as Jenny Lee mentioned, this is not just changes in the brain, but perhaps changes in the body as well, that could be either due to changes in the brain or due to roles that NMDA receptors play in the body. So these are some of the questions that I would like to answer, and would like to be able to give you good, solid responses to those questions. So there are a lot of reasons why we use mice in particular. And it might not make sense, for someone who isn't a researcher, why you would use these animals, because on the surface, they don't have a lot in common with humans. And so they're very practical reasons. At the heart of it is that as researchers working with humans, or working with animals, we have to find a model that is a good balance between approximating what's going on in the humans without working on animals that are sentient and that have you know, that are so close to humans that there's a lot of ethical concerns. So working with primates can be ethically very challenging. And so we try to look for what's called the lowest order species. So this is the kind of on the hierarchy of animals, with kind of us placing humans at the very top. We look for that animal that's as far away from humans that can still model humans. So this is what mice represent. And there's some other more practical reasons. They have very rapid reproductive life cycles. So mice are reproductively mature when they're 4 to 6 weeks old. And then they can have a new litter of mice every 3 weeks. And each litter of mice can have 6 to 8 mice, or sometimes as many as 10. So you can very quickly generate animals that have basically the same genetic background, and they're only different in the one gene, which is the gene that you're looking at. So this is really powerful, and there's not another mammal that, you know, that is better than mice in that sense. And they're also small, so that means that they don't cost a lot to feed them. You know, a larger animal, it needs more food, and they need more space. So that's a practical consideration. So those two things have meant that mice have a huge advantage over other animals, because we know so much now. We've used mice for so many years. Since the 50s, they've been our kind of workhorse animal. And so we're building on all of the knowledge that we have with every year of research that we do. And I just want to reassure you that in terms of GRIN, the gene is almost identical between mice and humans. So when we're looking at, for example, a point mutation that causes an amino acid change in a child, we can make the exact same change in the mice, and it's going to do the exact same thing in the NMDA receptor. So, you know, another consideration is that if we're ever going to find a therapy, one of the main model systems that's used for any therapy to be approved is a rodent, so either mice or rats, and then they do other testing in a non-rodent animal, like a dog, honestly, is usually what's used as the secondary one, to make sure that the drug isn't toxic before they would start doing anything in humans. So those are the reasons why we would use mice. However, there are some problems. And Jae-Sing, I think, captured that really nicely. They clearly have much simpler brains than humans, and they have very different behavior and physiology. What a mouse needs to do to survive is very different than what a human needs to do to survive. So, you know, they are nocturnal creatures. We are diurnal creatures. They only live for 2 years, and we can live for 80 to 100 years. You know, their metabolism is like 10 times faster than ours. They eat 10 times their body weight every day. So you can imagine that in all of this physiology, they are different. And so many times we will find drugs or therapies that work in mice, and then when we go move them to humans, they don't work. So I think that should be something that you keep in mind, that even if something looks really promising in mice, it doesn't always translate to humans. And in fact, there's a lot of frustration around making that transition from animals to humans. And also there's some aspects of human behavior that we just can't study in mice. So I'll give you an example. You know, some of your children have issues with reflux and vomiting, and that gag reflex isn't present in mice. So medicines that would cause nausea or vomiting aren't always picked up in mice, because they don't have that same behavior and physiology. So you know, other things like hallucinations. We don't know what the mice can see or what they can't see. So there's just many behaviors we just simply can't study in mice. Okay, so the other question that I think you may have is, you know, do we need to have a mouse made for my child's variant? And there's not an absolute answer, but I just want to give you some things to think about. Making a mouse is expensive. It's really not so much about the technology involved in making that an original mouse. It's more the time that it takes, and it can take about a year. And all of that is someone's salary and all the costs of doing animal experiments. And then it's all of the money that is involved in maintaining those mice for the years and years that it will take to study it. So we don't have endless resources to make, you know, 100 different flavors of mice. And I guess I want to reassure you that even though your child is perhaps unique and perhaps the only one who has a given variant, we are able to classify them and into certain categories. And so what we really need to do is make sure that we have each category of a variant covered. We don't necessarily need to make a mouse for every single amino acid substitution, or for a missense, I mean, for a nonsense mutation that would cause like a truncation mutation, or for just a big deletion. So those are some of the categories, you know, deletion versus missense. So deletions, we already have those mice, and those are the knockout mice that were made, you know, 10, 20 years ago, before we even knew about GRIN disorders. We were just studying NMDA receptors, and by we, I mean the scientific community. And so one of the ways that you study, you know, genes is to make a mouse that is lacking that gene. And so we have those that would correspond to the deletion variants that we see. And those are, most of those are available for us to just order. So it's those really, we don't necessarily need to be pushing for those. The missense ones, that's the one where we want to have mice that would represent, for example, gain or loss of function. So that would be the big category. But then we could also think about looking at different aspects of where, you know, like what part of the NMDA receptor is changed. Is it changed because of where it's located? Is it changed because it's not being activated with the neurotransmitter that it's supposed to be activated with? And so I would estimate that probably for each of the GRIN genes, we probably don't need more than, you know, 3 or 4 mice, and the knockouts, the deletion variants are really already out there. So we don't need to be making a lot more in order to get these categories covered. Because at the end of the day, what we want to do is find a therapy that's going to apply to as many kids as possible. We don't want to necessarily find a medicine that's only going to work for this one variant. And I think that it is possible for us to identify treatments that would be beneficial for lots of different types of variants that fall into these different categories. Okay, I wanted to kind of demonstrate how many mice we already have out there. So in kind of the big letters, I have the ones that that our lab is working on, and some of these we've had in the lab for a long, long time, and others we're just making now. So here's the status. This one we've been working with for about 20 years, and we know a lot about what this mouse looks like, how it behaves. medicines that make it better, medicines that make it worse. So we have a lot of information on these mice, and they represent mice that just have low levels of a wild type NMDA receptor. So they don't have changes in the spelling of the NMDA receptor, the GRIN1 gene. They just have low levels of the protein and the mRNA that's made. But we've been starting to make some of the patient variants, and you've probably heard me talk about this one. We've had this one in the lab for over a year now. This one we have had in the lab for a couple of months, and we're just starting to do experiments on that one. And this one is being made right now, and this one is still in the planning stages. So this is will be our first GRIN2B, but there's several other GRIN2Bs that are in the process of being made or have been made by other, I'm sorry, these are already existing right now. We have some GRIN2Ds. We have some other GRIN1s that other people have made. And, you know, I think right now, what we need to do is fill in some of these GRIN2As and think about maybe the GRIN3s or the GRIN2C. So I think that we're getting a pretty good list of mice, and the idea is that we'll be able to, you know, share these and for example, these two mice are being sent to Emory, and this one as well, for the Trainalis and Hongji Yuan's group. So we have you know, we'll be sharing these mice with anyone who wants to use them. So I wanted to spend the rest of my time with you to talk about, in more specifics, how we're using these mice to help people with a disorder. And, you know, I think the main advantage of mice is that we can look at their brain and in the live animal, and I mean, not the live animal, but we can either look at them while they're alive, or we can dissect out their brain after they die, and we can measure the levels of proteins and mRNA, the instructions to make these proteins, and we can look and see how specific parts of the brain are changed. So that gives us a lot of insight into what might be happening. The other thing that we can do is we can use these behavioral tests. And they're really strange, I think, to you know, to people who aren't mouse researchers. It doesn't make any sense. But let me just try and demystify this. Behavioral tests are kind of like the blood tests for how the brain is working. So if you have a problem with your liver, you would do a blood test, and they would see what are the levels of certain liver enzymes, and that would be the way to indicate, oh, it looks like the liver is not working properly. If you wanted to look at the heart, you would probably look at blood pressure and heart rate, and you'd maybe do electrocardiogram or something like that, and then you would get some indications about what's going on with the heart. Well, for the brain, a lot of how we know what's going on with the brain is to put these animals through these behavioral tests. And so this one we use a lot, where you just put the animal in a new place and you watch how it explores. And this can be used to understand specific circuits of the brain that are used in exploration of a new environment. And believe it or not, we know which circuits of the brain are involved in this. And you know, similarly, if we want to look at the circuits of the brain that are involved in social behavior, we would put them in a situation where they would have to choose, for example, between interacting with a mouse that they've never met before, or interacting with just the empty metal cage that the mouse is sitting in. And we try to put numbers on these things. So instead of saying, it looks like this mouse is not as social as this other mouse, we would try and have a test where we could put a number on it and say, this mouse spent 23 seconds here, and it spent, say 50 seconds in the empty cage. And so we can put a number on it. And then once you have a number, you can determine whether something makes an animal better or worse or closer to what a wild type mouse would look like. In this test, we're looking at how they deal in a stressful situation when they're out in the open. And so you're looking at how much time they spend out in the open versus how much time they spent in a safer, more enclosed environment. And the other one that I just want to point out is this thing called pre-pulse inhibition of acoustic startle response. And this seems like probably the one that would be farthest away from a human behavior. But in fact, it's measuring something that is very similar in humans. So here you see this mouse that's in a little cylinder, and in that box, we will play a sound, and it will be a loud sound, and the mouse will jump. And that startle response to a sound, we can measure exactly how high the mouse jumps. Humans do the same thing. We all know this. And so that's the acoustic startle response, and that can be measured. We can see how high does someone jump with a given acoustic sound. And we can also measure a circuit that is involved in dampening that response. And that circuit has to do with the arousal state of your brain, how alert you are, how much those circuits that are about watching out for danger, how much are those activated? And so if a mouse is in a highly alert state, then that mouse doesn't have the ability to attenuate its jump. And so it lacks that attenuation that can happen if you play a small sound just before you play the loud sound. And humans are the same way. And so if you play a soft sound right before the loud sound, then you'll get less of a jump. And that has to do with your brain's ability to filter things out. And so we can use this really strange behavior to assess certain brain circuits. And so even though this doesn't make any sense about why we would do these things or how this would have anything to do with your child's behavior, ultimately what these are just tests to try and get an idea of which aspects of the brain are not working properly, because we have a lot of knowledge about how certain behaviors correspond to certain brain regions or certain circuits of connections of brain regions together. And I want to finish up just by talking about some of how we've kind of put this all together. So we worked with the mice that just have low levels of NMDA receptors. And we did a lot of studies looking at their post-mortem brains. So we would measure the levels of proteins, the levels of mRNA, of gene transcripts. And the picture that we got from all of that detective work was they were showing signs of a bioenergetic deficit. So there were a lot of changes in mitochondrial genes. It looked like all of those mitochondrial genes were upregulated, like there was more mitochondria or they were working harder. And at the same time, we saw some other evidence that these mice were not absorbing glucose the way that they should. And so we started studying this aspect of how the mice are getting glucose into their brain. And glucose is the way that neurons like to get their energy. And the way that they do that is the glucose is floating here in the bloodstream. And glucose doesn't normally get into the brain. It needs to be actively moved into the brain. And it gets moved in first through these cells called astrocytes, and then the astrocytes feed the neurons. So the astrocytes are really important for giving that neuron the energy that it needs. And what we found out was that NMDA receptors are actually important in that process. And we're not the only ones. I mean, what I'm saying is that we found out by reading papers of other researchers who have demonstrated that NMDA receptors are important in this or can play a role in this. And so this movement of sugar, of glucose, through the astrocytes and into the neurons finally requires these things called glucose transporters. And NMDA receptors can regulate how well those glucose transporters are working. So we thought, oh dear, if this is not working properly because there are fewer NMDA receptors and these glucose transporters are not working, maybe we could provide an alternate energy source besides glucose that doesn't need to be transported in. And so we decided to try ketone esters, because when your body runs out of glucose, like when you're fasting, you haven't eaten for a long, long time, your body will start converting fat into ketones. And these ketones can be used by the brain as a different energy source. So this is the ketogenic diet, in the sense that when you take away all your sugar and your carbohydrates, then your body will use a different energy source, which is the ketones. And the ketones are fat, so they don't need to be actively moved through. They just can pass in to the neurons without any problem. And so we fed the animals a ketone ester, and we looked to see whether this would help with any of their behaviors and also what might be going on in the brain. So first, some of the behaviors. We put them in this open field here and let them wander around. And what you can see is that here we have what a wild type mouse looks like. And then if we feed the mouse the ketone ester, it really doesn't change their behavior at all. On the other hand, when we give the GRIN knockdown mice, when we put the GRIN knockdown mice in this environment, you can see that they're really hyperactive. This novelty kicks off their dopamine system, and dopamine facilitates movement. So they've got a lot of dopamine that's being released right now, and they run around quite a bit. And then what you also see is that over time, there's some what we call habituation, but it's not nearly to the level that it should be. So they're not really habituating over time as much as they should. And what we saw was what I would say is a modest improvement in this hyperactivity. So it was definitely lower, but this is not, we've certainly seen medicines that can work better. However, I just want to show you this, as this is kind of this is the treatment that we're working on right now. This is the newest data that I have. The other test that we did was this PPI of acoustic startle response, so that pre-pulse inhibition. And what you can see is that when you give pre-pulses of different intensities, you can get more and more inhibition of this startle. So these are wild types, and these are wild types treated with a beta hydroxybutyrate. And you can see that at this loudest pre-pulse, their startle response is reduced to about 60% of what it would be otherwise. And so here we see that the knockdown mice, oh, do I need to stop? Okay. Sandra, please let me know if I'm going over, and I'll stop. Okay.
No, just keep on going, Amy. You're fine.
Okay, thanks. Thanks, Christian. Okay, so what I wanted to show you is just that this is impaired in the knockdown mice, especially at these quieter decibels, and that it gets a little bit better with the beta hydroxybutyrate. In fact, it looks, almost like a wild type mouse. So maybe this is improving a little bit their behavior. This is not, maybe the miracle cure, but this is also not toxic. And this really isn't even the ketogenic diet. So this is just giving this extra supplement, food supplement, onto their normal diet. But what we could do with these mice, after we had run this battery of different behavioral tests, we can also then take their brains and study them. And so one of the things that we wanted to study was this idea of what's going on with the mitochondria. And so to do that, we take the living slice and we stain for mitochondrial activity. And this red glowing product is produced whenever the mitochondria are active. So the more red glow there is, the more active mitochondria. And I just want you to know, we're not looking at a single mitochondria here. This is actually a whole neuron here. So we're really zoomed way back, And we're looking at a population of neurons here. And what we're measuring is how bright are these neurons? How much fluorescence intensity is there? And so when we look at this, what we can see is there's a spectrum. So depending on the cell, there's more or less mitochondrial activity. And this doesn't really change that much with the BHB treatment in the wild types. But what you can see is that in the knockdown mice, there is slightly higher levels of mitochondrial activity. But what the BHB seems to do is to allow some of this increased mitochondrial activity. So the pattern is actually more spread out. And it seems that there's actually more energy production that's going on when you have this BHB as an energy source. And we did something called electron microscopy and really the reason why we did it is we wanted to look at the mitochondria. We wanted to look at how many there were, but we looked in a region of the brain where there's a lot of myelin, and myelin is the coating that goes around axons, and that's really important for the electrical signal to travel across the brain in a rapid way. And what you should see with myelin are these concentric rings that are tightly compacted, and there's a lot of proteins that hold those rings together. And so the more compacted you would see, that would be an indication that the myelin has good integrity. And so what we were surprised to see was some of the fields showed this non-compacted myelin, where you can see it's really ruffling. And we didn't see this everywhere in the brain, but in some of the sections we did see this. And then when we gave the GRIN knockdown mice the beta hydroxybutyrate, we saw less of this. So I'm not showing you the bar graphs, but I wanted to just give you some illustrations here to say that this aspect was improved and was surprising to us, because we were actually doing this study to look at mitochondria. But when we did this, we couldn't help but notice this other phenotype that we were not expecting to see. So so now to kind of put this together about where we are and trying to understand, it really is a detective story. It's a process of trying to understand what's going on. And so what we think is that normally sugar works very well to feed neurons, but in the knockdown mice who have low levels of NMDA receptor, that sugar is not a good energy source for them because they're not able to utilize it. It doesn't get into the neurons the way that it needs to. And so if you can supply them a different energy source, like a fat that doesn't need to be actively moved in, then they have the energy that they need to survive. But in the absence of this, you can have some neuron damage. If the neuron doesn't have enough energy, it could die. That could lead to an inflammatory response that happens whenever neurons are dying. And that inflammatory response actually leads to damage to the myelin. And so maybe what we're doing is just preventing that inflammation. And so these are some of the things that we want to do next. We do want to look at how this would affect EEG patterns and seizure frequency. We're now doing the ketogenic diet and BHB on the new GRIN variant mice, both the gain and loss of function. And we're going to do some more studies in how mitochondrial function is changed and how neuroinflammation might be contributing to that myelin phenotype. And I'll just finish by thanking all of my many collaborators. And especially I want to thank the parents and Cure GRIN for supporting our work and for giving us, the encouragement that we need to keep going. So I'll stop there and stop sharing my screen. Okay.
Thank you so much, Amy.
Sure.
That was beautiful. We have time for maybe a couple of questions for each one of you, Dr. Wen and you, and then we move into the roundtable discussion. We can do the rest of the questions. So I would start with Dr. Wen. There was a question from one of the families. It says, you have created organoids for GRIN. How much information do you have so far?
So we have generated many iPS cell lines from GRIN kits, and we are making organoids from them right now. But actually, unfortunately, due to the COVID-19, that stopped our capture for a couple months. But we have been wrapping up right now, so we are doing many test experiments right now. Actually, we are doing experiment similar to what Jenny Lee just mentioned, including the look at the neural development in the GRIN organoids, and we are looking at the neuronal functions, including the synaptic transmission and the EI balance in the GRIN organoids. And also, we are looking at the mitochondrial functions and the reactive oscillation. And as I mentioned, we are able to generate the microglia, and actually right now we are also looking at how the GRIN variants affect the neural microglia interaction and how this affect the neuronal functions. Yeah, but this is ongoing right now, but we don't have much primary data to present today.
Then another quick, probably just goes down the line. How do you have any compounds right now in your pipeline to test or screen with these GRIN organoids?
Yeah, we have several libraries in the lab, the small molecules, including, as I mentioned, the local library and the FDA approved drug and the clinical trial compounds. But we also have the chemical biology, the screening center that they also offer a different type of library. But our priority is on the synaptic modulator and some compound that are the mitochondrial modulator, and maybe we also will try some anti-inflammatory compounds. That's what we are looking for here.
Thank you. And the next ones are for Amy. How would you categorize the mouse that are already made? Are they considered a missense mutation?
Yes. I think that almost exclusively the new mice that have been made are missense mutations. So they have a substitution for one amino acid to another amino acid, and they're based on, patients. So we're not making them based on, there are some that were made like 10 years ago where they were saying, we think that this is where the glutamate binds and we're going to make this amino acid substitution to test that. But now the mice that are being made now are because there's a patient who has a specific variant. But the deletion variants, I think that people are not working on building those because we have the knockout that would give the same phenotype. And I guess the other one that we could think about building is something that is a nonsense, which means that it has a, instead of switching the amino acid, it has the message to stop. And so you get a partial protein that's made. And so we haven't really done those, but they might have been done in culture systems. And I think we could probably learn from the culture systems what is happening there. So by culture systems, I mean things like the work that Chavi Altafaj is doing, or Steve Trainellis' lab is doing, where they put them into HEK cells, or they put them into primary neurons, and they can see what happens there. So yeah, I think we have lots of loss of functions, and they honestly, in the mice, they look pretty similar. So, you know, I think this is another reason why it doesn't make too much sense to go crazy over making, you know, multiple variants. Is that from a therapeutic point of view, we can probably, we have enough. I think the one thing that we can't do is sometimes there have been situations where it's not really clear based on all of this cell data whether it would be a gain of function or a loss of function. And I think ultimately we're only going to get that from the mouse or maybe from the brain organoids. But I kind of think, Jae-Sing, I don't know, it seems like it's taking about the same amount of time for us to, you know, it's a long process.
Yeah.
And the last question for Amy, and then we move into the roundtable discussion. What is exploratory, non-exploratory behavior linked to in mice? Okay,
so the brain regions that are involved in exploratory behavior, we know this from, we can do lesion studies where you cause a small damage in a very small brain region. Or we can also do it with studies where you apply a chemical just to a very small region. And now they're doing these studies where they activate with lights, and there's special tools where you can activate specific pathways. So exploratory behavior is going to involve the front of your cortex, which is your kind of planning and movement part of your brain. It's going to involve your hippocampus, which is involved in remembering where you've been and what you've already explored, what you've seen, what you haven't seen. And it's also going to involve your dopamine system, because dopamine is needed for movement. And whenever you're in a new environment, your dopamine system increases, your dopamine firing increases a little bit, because your alertness level increases, because this is new. You don't know, it could be a very good thing, it could be a very bad thing. And so just in general, your arousal level increases. So that's what is happening during exploration. And then as you have explored something, if all of those systems are working properly, you will conclude that there's nothing more to explore. This area has been explored and there's really nothing here. And a mouse will generally go to sleep, because we do this during the day, which is their sleep cycle. But if your arousal system can't calm down, or if you can't recognize that you've already explored this, then you would continue to explore. So that would be where we would see this loss of habituation.